Everybody, it is a proud privilege to introduce one of the eminent scientists, Professor Apparao Kodile, before this learned gathering. Professor Kodile obtained MSc and also PhD from Sardar Patel University. Then he joined as a postdoctoral fellow at the Institute of Botany, Academia Sinica, Taipei, Taiwan, and subsequently was a visiting scientist, Institute of Molecular Biology, Academia Sinica, Taipei, Department of Plant Biochemistry and Biotechnology, University of Münster, Germany in 2006. Professor Kodile, basically he's a teacher and teaching general microbiology, molecular plant pathology, microbial technology, and microbial ecology to the PhD students. Professor Kodile's groups works on molecular plant microbe interactions and published about 85 research papers with special focus on recombinant bacterial chitinases, non-host-resistance nanotechnology for crop protection, and molecular interaction of rhizosphere bacteria with plants. This group is actively engaged in cloning and the characterization of bacterial chitinases. Professor Kodile is a member of International Research and Training Group on Molecular and Cellular Glycosciences between University of Hyderabad and University of Münster, Germany. And he is a recipient of several honors and awards. He has honored elected fellow of National Academy of Science, Agricultural Science, elected fellow of Association Microbiologist of India, member of DBT Task Force, member of Research Advisory Council, National Bureau of Agriculturally Important Microorganisms, associate editor of Australian Plant Pathology, elected fellow of the National Academy of Sciences, honorary fellow of Association of Biotechnology and Pharmacy, outstanding plant pathologist award by ISMPP in 2005, elected fellow of Andhra Pradesh Academy of Science and also Indian Phytopathological Society. Very recently, he was awarded the Rangoswamy Award 2013. It is a lifetime achievement award in microbiology by MI. With these words, may I request Dr. Apparao Kodile to interact with our learned audience about the plant immunity of plants.
Thank you, Professor Pardo. Good morning, everybody. It is indeed a great pleasure to be here at University of Kalyani for different reasons, I would say. The one important reason I would say is I got a chance to see a great name that we have a lot of admiration as a student, that is Professor Samadar. And I have learned Professor Shane developed this department of botany, and such type of a department to be here, to be part of you and to interact with you on something what I learned and what we are doing in Department of Plant Sciences at University of Hyderabad. It's a great pleasure for me. The second important reason I would say is that I'm here partly because your Vice Chancellor is from my university and he knows me closely and he wanted me to be here. You must know that this gentleman, your Vice Chancellor, is a committed individual. He doesn't have any other interest. He only wants anything that he does, he likes to do in a more meticulous manner and the best possible way. I'm sure the academicians here, the students here, the staff here are going to probably enjoy if you all want to, if you want to give your best to the university and develop your place. He would be the right man, and I request every one of you to take advantage of his presence and see that you grow one step further from the place where you are. I'm sure. I've gone through the profile of the department when I was interacting with Dr. Sudipto. I've seen there are good contributions coming from the department, and there is no limit to what is good. For you, University of Calcutta may look good. For me, University of Science may look good. For University of Science, MIT may look good, or Cambridge may look good. So there is no limit to what is good, better, and best, all that. But we have to, all that we have to think about is to move one step forward every time. And if we can take steps quicker, maybe we can grow faster. And if we take little slower, still we continue to grow. So let's remember that it is the best way to do, or the best way to grow would be to take steps forward and not stagnate at the place, nor take backward steps. So with these two points, I would like to, I'm also happy, I would say the third important reason, let me tell you, that a great professor like Professor Varshney is with you today to share his thoughts on protein synthesis. A well-known teacher, a well-known researcher, and I'm sure you're going to benefit from his talk and his experience on protein synthesis. With this little of introduction, I would like to switch on to the topic that I'm just click once. I would like, before that, I want to thank these three people who brought me here. Your Vice Chancellor, Professor Hanglu, Satanlal Hanglu, and Dr. Roy, who has taken all the pain. I think he has been in touch with me for the last 2 months. On constant basis, he has been talking to me. He is so hesitant that he would ask me every time whether that is the right time for him to talk, even if it is at 7:30 in the evening. He was so nice, and he made all his efforts to bring me here, and I must thank him for all his efforts. Of course, I would like to thank all the faculty of the Department of Botany who have unanimously thought of my name to be here, to be with you, and to share my experiences. Next one. Is there a way that we can, well, we may be able to manage. If we can switch off, it will be nice. I come from an agriculture background, and I come from a place where there is a lot of agriculture, and this is something what we see regularly in a place where agriculture is there. Even if there is no agriculture, we see a lot of plants, and our Dean rightly mentioned about the photosynthesis, the greenness of the plants, and how we depend on the plants, all that. Very nicely he has articulated that point. Such a beautiful nature, what we see here, the beautiful nature that we see around us is because of the plants. And these plants would not have survived. These plants would not have survived if they do not know how to fight against the pathogens. If they have no mechanism to resist the pathogens, they would not have survived, because we know there are thousands of fungi which can cause diseases of plants. There are hundreds of bacteria which can cause diseases of plants, and equally a good number of viruses which can cause diseases of plants. When there are plethora of this kind of pathogens around, we still find the plants green, plants healthy, and they're giving us the food, they're giving us everything that what we wanted. If we click once, this is what I imagine if the plants do not know how to fight against the pathogens. If they don't know how to fight against the pathogens and avoid these pathogens, all that there would not have been anything to see green. And the consequence of that is all of us would not have been here to have a seminar on plant sciences in genomics era. It would not be possible for us when there are no plants to be here. So there is something which is there in the plants which many people might not have thought in the early days, that they also know how to fight against the pathogens. A lot of things have come out in the recent past. More details of that, more science of it has come out in the recent past. Although we are all selfish when we talk about immunity in human beings, immune systems in human beings, a lot of things were done, and Nobel Prizes also were given. Nobel Prizes were also given for explaining the phenomena and how the antigenic variation, how the antibody variations are brought about by the mammalian systems. All that is known for quite some time. But unfortunately, the people who work on immunity in plants may not have the same recognition as much as we have recognition for those scientists, those researchers who work on immunity in animal systems, particularly the mammalian system. Next slide, please. I talk about the topic, and the plants have primarily one type of mechanism which many people believe is referred as non-host resistance, meaning the plants decide, saying that no to the plants, because they have interacted with them sufficiently, with these microorganisms, fungi, bacteria, viruses. They have interacted sufficiently for millions of years. They know what are all the different things that these organisms have, how do they behave, by what mechanisms, by what capabilities they are able to cause the disease. And when we know such type of organisms are there around, the plants have realized, well, what is the best way that I can keep them away from these plants, away from us. And that mechanism is operating at many different levels. I'm not going into details because it's not an exclusive topic on plant-pathogen interactions, but I would like to tell you that this is one way that the plants try to say no to the plant pathogens, that is non-host resistance. Many ways, at many different levels. If the pathogen is successful, it goes to the second level, third level, fourth level, like this. It goes on. But the plant has mechanisms to fail it every stage. And then if it fails at all these different stages, maybe there is the next level of resistance, what we call as host resistance, which we are, Professor Samuddar was asking me about, the gene-mediated resistance. That's the level at the final stage that you have. If that also fails, obviously the plant succumbs to the disease. That's where, my dear friends, my dear students, all of us know, disease is only an exception and not a rule in plant systems. Disease is only an exception. All that losses what we talk about are because of exceptions and because of the ingenuity of the pathogens. They try to dominate in certain situations and then take advantage of the environmental conditions and cause the disease. In my lab, we are interested, looks like we are interested in 4 different areas of research. I'm going to narrate to you about the plant immunity, taking my own journey in plant pathology. We are interested in 4 different areas of research, but back of my mind, I know what we are interested in, what is the best way to induce resistance in the plant and make the plants free from the disease. Natural mechanisms, natural resistance mechanisms of the plants, the immune system of the plant is that what we want to activate, and we want to see that the plants are free from the disease. Whether we talk about chitinases from bacterial sources, which I am going to talk more details in a little while from now, and non-host resistance that we work, a brief introduction, a brief outline of what we have done, I will present to you now. And I'm not going to talk anything about the plant growth promoting rhizobacteria that we work with, and the nanotechnology, application of the nanotechnology as to how we can make use of this technology for crop protection. Again, our intention is to induce resistance in the plant and not use any chemicals for the control of the plant disease. Next slide, please. Well, you may be wondering about the different things that I am showing here. What we see here is many of us are botany people. I'm sure more than 90% people sitting here are botanists. have background in botany and interested in botany, we know that this is a unique type of inflorescence which has its own, I mean, there can be 1,000 things I can show here. I just picked up a few. I picked up a few like this, and an animal like this, an animal like this, and some other plant examples, just to put some. What are we able to see here is, tomorrow if somebody shows me this kind of a structure, immediately we will say it's banana. And we will never miss this inflorescence of Calotropis. We will not, I might have already done this way back in my BSc or sometime, but I will never miss that. This is an inflorescence of Calotropis because of its unique appearance. And the morphology of the leaf is unique. And if you see the arrangement of the seeds in our pomegranate, it has a pattern of itself. What am I trying to drive? I'm trying to drive that if we see this second time, we know it is copra. It has a pattern, a unique pattern of itself. A unique pattern of itself which we will never be able to forget. And such type of patterns, when we are able to recognize, don't you think that the plants have the capability to recognize the patterns? We must agree that the plants also have a mechanism to recognize such kind of patterns. We are recognizing these patterns because we are able to see with our eyes. Even if I don't see it with my eyes, tomorrow if somebody gives me a fruit of this kind, by touching this fruit from one end to the other end, by looking at the shape of it and by the texture of it, I would like to say that it is banana. I would conclude that it is banana, even if I am not able to see that. Even if I don't see that, I will still call it banana because the pattern is so unique. So if plants cannot see with their eyes, they can at least perceive. They can recognize that pattern. Click please. Such kind of patterns recognition of these are pathogen associated molecular patterns which are recognized. The plants have a mechanism to recognize a pattern which is unique to a pathogen, to a group of pathogens, a large group of pathogens like fungi. What is that the fungus has on the surface? All of them have a cell wall. The first thing that they come in contact with the plant, when it is touching the plant surface, the molecule that is interacting with the plant is fungal cell wall. Similarly, if we are talking about a bacterial pathogen, we are talking about a peptidoglycan, the cell wall of the bacterium. It is these molecules of the surface which are in direct contact with the plant which are being recognized. We call them as pathogen-associated molecular patterns. And these pathogen-associated molecular patterns are known to trigger immunity in plants. You just have to see something. This is a pattern of the fungus which is recognized in the form of chitin or an oligomer of chitin. A pattern of the bacterium which is recognized as a peptidoglycan molecule. And what we see is, interestingly, they have some unique receptors on the plant membrane, unique receptors, unique proteinaceous molecules on the membrane of the plant which can recognize, which can interact with these molecules. They are typically defined as molecules which have repeated units of lysine motifs. Lysine motifs we call them. There may be 2, there may be 3 lysine motifs, but they have lysine motifs. When they have lysine motifs, they have the capability to interact with carbohydrate polygomers, carbohydrate molecules. What are the carbohydrate molecules here? The carbohydrate molecules are coming from the pathogen. That is the fungus in the form of cell wall, maybe a peptidoglycan in the form of for a bacterium. And if you see that, a rhizobium, which also enters into a symbiosis, also has a molecule which is recognized again by the same kind of lysine domain molecules which have lysine domains. But then the outcome of interaction here is nodulation. The outcome of interaction is resistance here. When it is interacting with a fungal molecule or a bacterial pathogen molecule, it results into kind of resistance, whereas in case of a rhizobium when it is interacting with another carbohydrate molecule, lipid chitin oligosaccharide. Lipid chitin oligosaccharide, which is again primarily a carbohydrate which has a lipid moiety attached to it. Next slide, please. As plant scientists, we may be talking about this now, but you look at this, variety of these kind of receptors are identified in mammalian systems. Variety of these mammalian receptors are identified in the mammalian systems in the membrane. And these pathogen-associated molecular patterns from the pathogens are also known in animal systems. Animals also suffer from diseases. They are also caused by bacteria. They are also caused by fungi. And such type of molecules of pathogen origin are known to interact, and they are known to activate different kind of immune responses in animal systems. It's not something new that we are talking. Only thing is we come to know about these receptors, these unique patterns that are there on the pathogens. Therefore, this kind of mechanism, since they are recognizing pathogen-associated molecular patterns, these receptors are designated as pattern recognition receptors, or PRRs. Pattern recognition receptors, or PRRs. Next slide, please. So now we see that there are basic principles of plant immunity I talk about. A pathogen is interacting with the plant and releases pathogen-associated molecular patterns which can be recognized by the pattern recognition receptors, and they activate, through a kinase domain, they activate, there is a transmembrane domain which translates the message, and then the kinase is activated, leads to cellular signaling and an immune response. The plant will be free from disease. But then the pathogen will not keep quiet. If the plant is going to express its response by recognizing the pattern of the pathogen is not going to keep quiet. And pathogen, what it does is it secretes or injects. In the case of bacteria, it is very well known through a kind of a type 3 secretion system, we call it. They inject the molecules into the plant cell, and fungi send these molecules through, some of the biotrophic fungi send these molecules through haustoria. They release these molecules, these effector molecules, into the plant cell. These molecules, when they are released, the purpose of these molecules is to interfere with the signal that is generated by the interaction of the pathogen-associated molecular pattern and that of the PRR. The signal that is coming is being interfered by the effector molecules that are released. Then there will be no response. There is no immune response here because it is interfered. That leads to what you call as effector-triggered susceptibility in the plant. The third step, well, the plant realizes now that there are some effectors which are doing this kind of a job. The plants can have another pattern recognition receptor inside the cell now. These are located extracellularly and these are intracellularly. These pattern recognition receptors, which are inside the cell now, can interact with, can find and negate the effect of these effectors, and then can allow this cellular signaling or activate another signal pathway which leads to immune response in the plant. So we have a wonderful mechanism, by a molecular mechanism, in which the plant immunity is being operated. This is the basic level at which it is operated. When the pathogen has these effectors, you may find this kind of additional specialized molecules which are formed. These are more general because this is the fungal cell wall. For several of the fungi, the chitin is the cell wall. For several of the bacteria, peptidoglycan is the component of the cell wall. Whereas these effectors are unique for a pathogen. So when an effector molecule is very specific from the pathogen, the receptor also will be very specific to that. Therefore, you find a specificity in this kind of an interaction on R and AVR gene interactions. That's what we see, resulting in effector-triggered immunity in plants again. Next slide, please. Now I switch on to, I have given you a basic introduction of what I mean by immunity in plants. I would like to switch over to a little of what we have been doing, share some of our experiences on this immunity in plants. The first thing that we have been working on is non-host resistance in the plants. non-host resistance in the plants. We have, I have two bright students who joined after both of them were toppers of the batch. They wanted to do research and they approached me. And I said whether if they would be interested to work in that. They read the literature and started working on this non-host resistance. I suggested to them that they should think about two alternative model systems if they can see. And one of them has devised one model system in a plant and a bacterium interaction. Other one devised a model system, plant and a fungus interaction. And before that, we of course contributed. Our whole idea was that there are molecules at the surface, that is particularly on the extracellular matrix and the plasma membrane continuum. There are molecules which can interact with the molecules from the pathogen and then can suppress the pathogens. That is what is our idea of non-host resistance, with which we went on and we categorized. These are type 1 and type 2 type of non-host resistances are known, and we contributed a review on this and in Journal of Plant Physiology and went on to do some work. I'll share briefly about little of that work. Next one, please. This is first model system, which is done by a student by name Swaruparani. I'm going to show her a picture later. Rice is known to cause disease. Xanthomonas oryzae can cause disease on rice, whereas Xanthomonas citri cannot cause disease on rice. We know that. There is a host specificity there. Citri can cause disease on citrus, that is citrus canker, what we see here, and whereas Xanthomonas oryzae cannot. So this will be a non-pathogen on this. Whereas citrus is a non-host for this pathogen, rice will be a non-host for this pathogen. When there is a disease, possibility of disease, we are referring it as compatible interaction. When there is no possibility of disease, we are referring it as incompatible interaction, a non-host interaction. Next slide, please. What Swarupa has asked is the questions that what happens upon inoculation of Xanthomonas citrus onto the leaves? What are the different anatomical and biochemical changes that occur during the citrus and Xanthomonas interaction? That is in a host interaction. And Xanthomonas, when I say we are talking about either with Xanthomonas citri, Xanthomonas axonopodis citri, ZAC we call it, or Xanthomonas oryzae, Pathovar oryzae, we call it a zoo. Either with the ZAC and zoo, in these interactions, what happens? Which proteins are differentially expressed in this kind of interaction when it is compatible and incompatible? And what are the extracellular matrix associated protein changes that occur in citrus Xanthomonas interaction, both in compatible and non-host interaction? And what are the nuclear proteins that change during this interaction? We thought that this will help us to understand comprehensively about what are the different kinds of changes that are occurring in plant and bacterial interaction when it is compatible and incompatible by comparing those two systems. Next slide, please. This is just one slide to show you that this is a Xanthomonas axonopodis citri where disease develops. Oryzae, when you inoculate, disease does not develop. You find that the suppression of the disease probably is due to the hypersensitive reaction, what we see when it is interacting with a non-host, and which is evidenced by reactive oxygen species and deposition of callose. Some of these are active defense responses that are known to be operating against the pathogens. We don't find those things in when it is interacting with its host, and therefore there is development of disease. Next slide. Well, this is just to show you a picture of how we devised our model. We try to isolate the wall-bound proteins, the proteins from the wall-bound, and those which are soluble from the extracellular matrix, soluble proteins, and against a ZAC challenge and against a zoo challenge. These are the proteins. That's how we devised that experiment. Next slide, please. This is how one would go do a 2D proteomic study. We have to have biological triplicates. We have to follow basic rules of proteomics before we end upon. I'm particularly happy that I'm talking about it when you have my title your seminar as Plant Sciences in Genomic Era. Next slide, please. Just a glimpse of a little of what she has observed. You will find in Xanthomonas axonopodis citri interaction, these are
The proteins which are upregulated, you can see in blue here, the soluble extracellular matrix and those which are associated